Hello, all. John Quinn, Director of Science and Product Development for Flojo, and today we are going to do the BD CellView Lens. Let me share my screen, put up some slides. We'll start in about a minute. It is still 12:59 my time. Here we go. Okay. But yes, just to do housekeeping, I will keep the chat open the entire webinar so that I can see any questions that come in or discussion that happens. We have a separate, I guess we've got QA also, but I kind of like the chat. It's a little quicker and easier to make use of. So if you're good with that, I'm good with that. Um, my email's right there on the screen, john.flojo@bd.com. So if you want to reach me for any, follow-up or questions or discussion, that's the best thing to use. And with that, it's 1 o'clock, so why don't we get going? Welcome to the Flojo webinar series. As I mentioned, this month's advanced is the BD CellView Lens plugin. My plan is to subject you to something like 15 slides, and then we'll open up the software, and I'll actually just demo how to do everything that I tell you you can do during the slideshow. Okay, so plan for the webinar. We will walk through extracting TIFF files, adjusting image filters, population selection, creating image sets, which I think are a really nice way to make your work with the images real actionable, and then using HyperFinder to recapitulate whatever set of cells that you'd like to, make gates on, take forward into HyperFinder, or sorry, take forward into Chorus for sorting or something like that. So the tool that we're using today is version 2.0.15. We posted that one, the latest update on April 11th to the Flojo Exchange, which is off the just flojo.com website. To get the plugin, you hit the download button off of the CellView Lens tile. We recommend using this one with Flojo 10.10, which has been out for a while. So that's probably, if you've got Flojo 10, it is probably what you're already using. And I will show you the bits of it once we get out of the slides. Okay, so step one. How do we get image files? So off of Chorus, the image files are compressed into CoreWave files, .cvw. You'll need to extract those for Flojo to use them into TIFF files, which are what's compressed into that big CoreWave file. The CoreWave file is real nice in that it's massively compacted, so it's much easier to port than thousands and thousands of TIFF files. But again, we have to extract them at some point. You can do it using the BD CellView Lens image extractor, which is a separate tool that comes with Chorus. It is, I believe, PC only still at the moment. So if you're in a Mac, you'll want to use one of the other two options. If you store the data in the BD Research Cloud, the BD Research Cloud has a built-in image extractor, so you can upload a CVW, point to it on the Exchange, and extract the files up there. You can also extract the files directly in the CellView Lens plugin. So if you put the CVW file on your computer in the same place as where the FCS file is, when you start the CellView Lens plugin, it'll just ask if you'd like to extract it, and it's pretty quick there. So any of those options are fair game. Do whichever is easiest for you. We will do the bottom one in today's webinar since we're focusing on the CellView Lens plugin. Okay. The next thing you might like to do once you start up the plugin is do some image filter adjustment. And so the basic gist of this is that we'll give you a preview of what you're going to see up here. Some filters where you can move the sliders up and down to adjust the thresholds. We will have gotten the image filter set from Chorus written into the FCS file when you get it. So it should look pretty much like what you left Chorus with. You may have to rename the parameters down here if you like. We will use like the detectors and all the bandpass and stuff. So if you just want to call it like DNA die or whatever, you can type in a name. You've got controls over here for which parameters do you want to show at any given time, and ways to adjust the masking down here that I'll go through once we have the software open. Of some importance is that there's also some automation we've built in for removing background, finding the best threshold for where we'd like to separate background from actual collected signal, whether to use a mask on all the channels or just the selected channel and so forth. So we'll cover that in the software, but this is the main tool for I'd like to adjust the images so they look as nice as possible. Okay, after that, it comes to identifying populations. You will want to identify particular populations of cells to look at the images of those, or to sort down like in a much more general way to some larger subset of good, bad, and different, whatever. You can use any tool that's in FlowJo. And so if you want to use a set of standard gates, that's fine. A clustering algorithm, that's fine. It's all good. Essentially, you just either apply on a specific population the CellView Lens plugin or drag and drop a node to something like to look at. So here, even if I just have, say, gated on scatter, and I've got things that I would call poor based on scatter and good, maybe singlets versus doublets, something like that. You can drag a, just in one of these icons, the node for the plugin to either one, and open up a graph out of the CellView Lens plugin and be able to mouse over the images to see like what's the difference between poor and good scatter. And then open up the entire image wall to say, okay, so the good ones look like I've got a lot of single cells. The bad ones have got a lot of doublets. Bad being very subjective here. There will definitely be experiments where you're looking to find, two cells talking together and so forth. But that's the basic gist of it. And then there's also a sort icon for sorting uh, based on any parameter you want, ascending or descending, so that you organize the data based on the thing that's most interesting to you. And also just to mention here that you can use any of the tools in FlowJo, so such as like you can make t-SNE plots. So if you wanted to look at any of the parameters, or sorry, some bulk set of parameters holistically, and then use the mouse over and image tools, you can do that as well. So we can go in there and we see like what specific islands within the t-SNE plot might look like. Okay. And then I've kept in, every time I do one of these talks, I keep in the slides that are just what are the image parameters? Just because I feel like that is one of the primary questions that are worth knowing. I'm curious if that's over the slide for you all also. It might be the picture of me talking. I'll move that out the way. I think the most important thing to remember is that we will calculate all of these different factors of like how round or how correlated, etc., are parameters for every one of the parameters that are off of the image detectors. So there's 6 enabled blue laser detectors, and here they are. And every one of the features, so radial moment, max intensity, and so forth, are calculated for all of them. So you can get the radial moment of side scatter, the radial moment of channel 2, etc. And so what are those? Eccentricity. So for whichever parameters you're calculating, so this picture almost looks like one where you might imagine we're calculating the eccentricity of, say, something like a DNA dye. But you could do it of scatter or something like that to get the measure of the entire cell. It's essentially how round versus oblong your signal is. And so here's a histogram showing low to high eccentricity and some pictures from the low region versus the high region. And so eccentricity might be something you could help to identify singlets versus doublets, or one cell versus one cell versus two, which is singlets versus doublets. So anyway, yeah, or two cells talking to each other, or things like that. So that's eccentricity. Moving my chat around so I can see the whole screen. Max intensity, just what is the brightest single spot, on that particular cell. And so measuring, the brightest versus the total might be a way to see, some differences. And do I have a lot of signal, but it's all concentrated versus a more just dispersed signal? So that one's pretty straightforward. The brightest ones are really bright. The dim ones are dim. Size, basically just your pixel coverage. Do you have more of the area of the cell covered with signal versus less? So are these bigger cells or smaller cells? Radial moment. Is your signal actually, more concentrated in the middle or spread like a radius to the edges? And so again, the histogram of low versus high. So concentrated versus more spread toward the edges. Correlation, if you've got two fluorescent channels, where you can see, is their expression correlated? Do two things tend to track together? Delta center of mass, the distance between two different parameters. So again, do these track together? An interesting one for, again, if you've got cells talking, do two different types of cells tend to line up? And are they tight together talking, farther apart, and so forth? Diffusivity, do you have a lot of punctate fluorescence or more evenly spread fluorescence. So if you're looking for some kind of particles, some kind of maybe like phagocytosis experiment might show a lot of diffusivity of a particular particle. Okay, so that's the kind of parameters. I went through that quickly. Hopefully that's okay. And we'll use some of them when we actually go through the data, and so that'll reinforce. One other thing that I just wanted to mention before we get actually into opening the software is that FlowJo has scaling preferences for every cytometer. So for my FACSDiscover S8, for example, I've used these to do things like hide the T-W or -H version of each of the parameters, except for a few and nots like SC-H if I'd like to use side scatter -H to help me figure out single cells or something like that. And nice to shorten your parameter list to just the ones that you're actually going to work with. I find I don't use the T's, W's, or H's very much. There's also an advanced rules button down here that will bring up this dialog where you can set specific scales on an individual parameter basis if you want that are remembered as preferences. So if you do it one time, you never have to do it again. Every time you open up a file in FlowJo, everything will be scaled how you like it to be. On our website, we've got some recommendations for the particular scale for each one of the image drive parameters and everything else that comes off the S8. It's just on flowjo.com documentation. And it's nice in that you will actually get a graphical preview of what it is that you're adjusting and can just adjust it till it looks right and then hit go. Or okay, as the case may be. So anyway, so my data should look okay when we start working with it, mostly because I've set my preferences for the S8. Okay, well, there's the blow up of the up over there is filtering. Okay, I'm going to talk about image sets during this presentation as well. At some point, you may draw some kind of gate on your data and identify a whole bunch of cells that look mostly just what you're looking for, or even some subset looks like an interesting type of cell within that larger gate. You can click the Show Image Sets button and start manually curating specific images that look right, that look like what you were aiming for. And there's a tool in here for find more of these using a nearest neighbor algorithm. So you can essentially define a handful of exemplars and go ask the machine within a larger gate to go find the particular cells that are doing the thing that you are looking for to see if they're doing. Okay. And then we can use HyperFinder. I'll show all this in the software where you can define which parameters are fair game, which population it is that you would like to replicate. And HyperFinder will build a series of gates that will produce the same outcome as any path that you got to it, whether it was clustering or in this case, I might show you using image sets and picking a few events and showing what kind of gate would find those events. They become gates in the FlowJo workspace. And then if you'd like, okay, well, there we go. It's making gates in the FlowJo workspace. And then if you'd like, you can open a FlowJo workspace back up in Chorus and actually sort on any gates, including the ones created by HyperFinder. So that makes this totally actionable, that if you find some things you like, HyperFinder can give you a gating strategy to get more of them or to isolate them or collect them or sort them, and then you can go do it. So that is what I'll show you how to do in the software. That was the quick overview. I see no questions in the chat at the moment, so let's get to it. Ah, yes, sorry. There was one more animation. There's an import button. All right, and now it is done with them. Okay, so, and I might, whatever. I was gonna say I might turn off my I think I might turn off my camera so my video stops getting in the way of things you're seeing. All right, this is the end of looking at me, which will probably improve the rest of the webinar. There we go. Boom. Okay. My friend Aaron Middlebrook collected some cell cycle data that I'm going to use today. I've extracted this and done some downstream work before, but I want to be able to show you how to extract. So I'm going to move my extracted images to the trash and just start from scratch as if we just have an FCS file and a CVW file. And I'm going to drag that FCS file into FlowJo. It'll pop up. There we go. And step one, let us go to Workspace and Plugins and run the BD CellView Lens. We'll be asked to save our workspace as one of the first things we do almost all the time. We'll call that CellView Lens _demo. Okay. And then it should, hold on a second, maybe I need to empty the trash to make sure those images are gone. And then start the CellView Lens. Okay, or let me start a new workspace and do that with the trash emptied. You can see how that I did this earlier, and so I've probably fouled up this is the first time that you've extracted the bin. All right, let's try that again. And that's still emptying out the trash right now. Lots of pictures. Boom. Okay. Call number two. Okay. Exciting. And I emptied the trash. So I've actually destroyed the images. So this one probably will not still have them. All right, technical glitch. Let's try this again from scratch. All right, FCS file in. No images. Okay, and that's what I was expecting to see before I emptied the trash. Would you like to extract these images? I say yes. You will see the folder gets created here that's got the same name as the FCS file. It says extracting, and it takes about 5 seconds to do, however many images we have here. There's 250,000 events. I think roughly half of them have images, and I will show you how to figure that out exactly. Okay, but step one, let us then move into our image filters. So you'll notice that the filter set added by default down here matches the name of the file. There's a few other defaults you can use, but this is the one that'll actually have the correct parameters that you actually used in the course experiment selected, and basically your colors and so forth the same. So anyway, I am going to open up that image filter display that I showed in the slideshow. And here we go. So we've got two fluorescent colors in here. One is a DNA dye, and the other is a membrane dye. That's a very dark color. Just call it membrane, close enough. And maybe we'll switch that. Let's make that like a bright pink or something. So there we go. And there's our overlay of pink and green. At the moment, we are normalizing everything in this preview window, the graph of am I looking at how much signal to the light loss channel. So for light loss, we'll be able to see the graph move and so forth. And so that looks kind of nice. And you can adjust, what these images look like by switching up what the minimum level of background is. The gamma is essentially the saturation amount, and you can adjust that as you will, whether you like it smoothed or not, and what you cap the maximum level out at. So you can do that for any parameter by clicking on that parameter. Unfortunately, they're normalized to light loss signal, which is a lot brighter. So the rest of the signals look really small and they're hard to see, but the controls work the same, where you can adjust these to get rid of background signal and make sure you keep it bright. So this is where a biological control is nice. You may want to use a sample that you don't expect to actually have the dyes, and to make sure that you set the threshold so those samples look blank. And then when you put on a positive control or something that may express the dyes, if you see signal, then you'll know it's a real level of signal. One node of the platform dragged to any other population or any other sample, and then even if adjusted, will be common throughout the experiment. So if you do set your thresholds on a control, then they will apply to everything else that you want to look at it with. Okay, turning on or off a parameter is just by double clicking, or you can see that these boxes over here serve the same purpose. So here we're getting a preview that will have, two colors and the light loss channel. We could turn off light loss if we just want to see like the two colors, and maybe I'll go to the green channel and take off a little bit of background. Okay, cool. So you can also use the automatic remove background. If I check this button here, it's going to use whatever parameter we have selected as the masking parameter, indicated with this little mask icon. And you can add multiple parameters by hitting the Edit Regions mask channel and clicking multiple channels to use for creating where the mask should be. Maybe the membrane's a good one. I don't know. And then also, if you're in any one of these, then the adjustments here are going to be for, we can show the mask region if we want to. That's kind of a fun thing. Um, if you've got edit mask selected, then this will actually, you can see, be a slider for how big the mask is. I just want to put it on one channel to emphasize exactly what we're doing, but I do like using multiple channels to figure out the best mask. Okay, maybe turn off the mask region. Okay, so you can adjust it. There's also a find best threshold where it'll go in and it'll try to automatically detect where the edge of the cells are. The last option up here, mask all channels. Typically what we're going to do is mask the overlay, but if you also want to see the same amount of signal in the individual channels that you do see in the overlay, turn that one on. And so I think that's generally a nice one to have put on. Okay. So I think that's the most important part of this. You can also save or import filter sets if you do the same kind of experiment multiple times. You want it to like look exactly the same with the names put in and all that stuff. There's a few different kinds of overlays, but the blended one's by far and away the best. So I think that is what you might do in terms of modifying your images. Let's apply the filters to the workspace. Okay, and you see everything switched up in the background. I can close this window. And we're now looking at an image wall here composed of all the cells in the experiment that have pictures with that particular image set filter applied. A few other things that I want to point out here before we do anything else. So we're in wall view now. So that shows as many individual cells using whichever, oh, I didn't show the button, whichever channel the eyeball is on. So we could switch it to membrane, and then we would be just looking at the membranes. Or we can go to channel view, where we break down each of the individual channels, including then the overlay channel. You recall, the plus adds rows, the minus subtracts rows, previous, next, advances to the next group of however many things you've got shown. Here it'd be 20. If we go to the wall view, it'd be like 50 something. So we could hit the next arrow and be on to the next group of events. Okay, so there's that. If we want to look at where these events fall on a 2D plot, I can hit the 2D plot button. It brings up whatever graph I was last looking at. You'll see there's a yellow line around the edge. And then when you mouse into the plot, it becomes active and you see this becomes a green line. And so you can mouse over it. You see some pictures. You'll notice that if you say missing image at this point, those are, we didn't collect every single picture of every single event. And I will show you how to isolate the ones we have pictures for. This works reverse. Also, you can click on a particular event and it pops up on that graph, which is behind this picture now. So let me move that. All right. And it is stuck behind my zoom control. So let me stretch it, then move it. Sorry for the enormous graph. Okay. And it'll work a little better if it's on the side. And I can just click and we can watch where the cross goes. Okay. We can also sort these images. And so you could say something like, well, right now they're just a number of events. You could go max intensity neon green, and then a sort ascending or descending. So here there's some bits that are probably just loose nucleus. We probably might want to gate those out. You could do like eccentricity of neon green, which is our DNA dye. And then we'll see the ones that are more perfectly round versus more stretched out. And so there's some extreme ones in here because we haven't done any gating whatsoever. But I really like the sorting tool for highlighting the cells where you want. You can also, at any point, if you decide you've sorted and everything below or above a line is what you want in a particular gate, there is under tools the ability to create a bisect gate, which uses that event as the threshold for I want everything right here separated into two gates, and you can pick the one which is the one you like. Okay, other controls. So if I've used find best threshold, Flojo goes in and tries to identify edges. And so we can actually take advantage of this and show regions. And so right now, almost everything is one region, but here's, one particular event that was found as two. You can scroll through and you'll see here's one that was actually found as three. And so there are a series of different regions identified here. Not super interesting in this experiment, but there'll be a lot of experiments where you might want to see cells interacting and you might want to work with that. I will mention there is a tool for calculate image parameters. So that will create a whole bunch of parameters that are like the distance between regions, the number of regions, and things like that. I'm not going to hit it right now. It takes 5 to 10 minutes to run because it's going to now have to go through and actually calculate all the regions and then a bunch of stats on them. But I've got a second workspace opened up in the background where I did that earlier. Oops, I did close everything because I broke it when I was deleting the images. I will reopen this workspace here that has calculated images in it when we get through looking at this. And so you can actually see them used. Okay, so that's the regions. I am going to turn back on light loss so that I can show the next couple, which are edges. So edges are a bit easier to see if you've got the light loss on. The connections, so the distance from the closest point of each region to the next, which are again, that distance is something we can calculate. You could choose to show the centroids as well if you care to. So you've got all of those tools as options. And now I'm going to turn back on off light loss, I think. Kind of like the black image myself, the black background. Up to you. Okay, so there's that. Let's do a tools and make image files gate now, which is going to make a derived parameter that says this is a file that has an image versus this is a file that doesn't have an image. And then we'll see that if we drop our CellView Lens plugin into the two populations, the one with a 1 is the one that has all the pictures, and the one with the 0 are all the events that have no pictures. Oops, did I hit the 1 again? Well, that should be empty. Interesting. All right, well, let us open up this guy here. And you'll see that this one has essentially everything we've done so far, but it also, I hit the go button on calculating your various image parameters so you can see what you get. The average distance between subregions. So if there's more than one region in a file, that's it. This is the image file parameter. It was the 0, the 1 there. Um, how many subregions there are, number of them, the mask area, the mask circumference, and so forth. And these are all just available for any kind of gating you want. So if you want to do like, I don't know, number of subregions, we can do that. And it looks like in this data file, it's largely 0 or 1. There's a blip at 2 where there's a tiny amount. There's probably even smaller amount at 3. Let's customize this axis a little bit just to go below 0 to really see that peak. Okay, and then I could do something like create gates on peaks. All right, there's a tiny number with 2, 3, 4, 5, or 6 regions. But if we want it, then we can gate on the ones with exactly 1 subregion or 2 subregions and so forth. A whole bunch of 0 subregions, which are the events without any pictures. Okay, yeah, so actually 1,500 which is 0.62%, but with 3 regions, and then a lot less after that. So there's a way just to figure out, for example, if you want to do 2 cells in a particular file that were maybe talking to each other, something like that. All right, I've put 1 gate on this data. I just did light loss versus side scatter, just to grab things that maybe look like more whole cells as opposed to that smear up there. And the rest of it we can do with the CellView Lens plugin. Okay, and then that picture there is eccentricity of side scatter versus radial moment. I might actually zoom in particularly on eccentricity of neon green, which is our DNA dye, and these may look very similar. Radial moment of neon green. Yeah, okay. So the scatter and the radial moment kind of went together for neon green. sorry, neon green versus NSIDE scatter were similar for their eccentricities and radial moments. So longish cells, in terms of their DNA, tended to be longish for the actual area taken up by the scatter as well, which is probably dominated by the DNA. So, sense. Okay, so we've got probably single cells and then probably double cells, or cells doing something more interesting. And since this is a cell cycle experiment, we could imagine something like we could gate the weirder ones and maybe hope to see a bunch of like telophase and anaphase. So we'll say these are some overall kind of M phase. And let's drag a CellView node, lens node onto those. Okay, and I want to turn off the regions, the edges, the connections, and the centroids. Oops, I keep doing that, focusing on that. So I want to actually just turn it off and look at the overlay. Okay, so there we go. We've got some cool stuff in there, or we've definitely got one cell on the verge of becoming two, or the DNA splitting, like we might have more anaphase there. And so that's kind of cool. That's an interesting one. Now, imagining that there was a particular condition that we were looking for, this is where we might want to use some image sets. So I'm going to hit show image set. Okay. It's right now just called images. I'm going to unlock it. Maybe I'll rename that as, let's call it anaphase. Okay. And here I can just click on cells that look to me like we've got kind of straight lines in the DNA and some membrane spread on both sides of it. And I keep moving this like Flojo speaking all over the place. And you can see the green box is highlighting the ones that I'm picking. I'm getting the pictures into my list right there. Do, do, do. Okay, so there's a few, 3, 4, 5, 6, 7, 9, 10. One interesting bit here is that I can pull this image here. Okay, so here's my plot of what it is I'm looking for. All right, and you'll notice that these buttons lit up, and if I click one, it's going to open up the layout editor with a blowout of here are the selected cells from my over here from my blah, blah, blah image set, and a line pointing to here's the actual image and here's the location it came from. So I could say to myself, well, it looks like if I had gated on the lower portion of this, I tend to get more of these kind of cells. And we could try out what the higher portion is. Maybe that's going to be more like our telophase cells or something to that effect. The other button just organizes it a little differently. Instead of by quadrants, kind of just pushes it like around the plot. So whatever formatting you like is fine. Okay, so I've collected a handful of these. It's a relatively large population, and I want to pick out cells that look like this. What I can do to boost up this particular image set is hit the nearest neighbors button. Find similar images using nearest neighbors. So if I click that, it brings up this list and says, which parameters would you like to use to identify similar cells based exclusively on the numbers? So right off the bat, I like that 147, the membrane dye, and Lexaflor, PS103 are highlighted. Stone 3, we could do DAPI, we could do neon green. So we could use those measures that are actually in the fluorescent data to say, find me similar things. But then we could also go and say things like, well, find things that have a similar size of that neon green in the images of neon green, and find me things with similar max intensity. And we can do long and short axis moment as well. Basically, to me, anything that tells me something about the shape of the neon green of the DNA dye is going to be useful, because whether it's farther apart, whether it's roundish, whether it's spread out in both axes or one, everything that looks like this is going to be measured with some of these. The center of mass might be less interesting, but we can do total intensity, radial moment. Could have probably used the membrane dye as well. Maybe we'll do the radial moment of the CD147. I don't think I care too much about the total intensity of the membrane dye. If it got a little more, a little less, that's fine by me. The diffusivity of the neon green is probably good. And then for whatever reason, we also by default highlight some of the metadata. I'm going to turn those off actually. Those are not going to be helpful. And we also highlight the repeat SW unmixed parameters. I'm going to turn them off as well. Okay. You can tell it how many you want to find. So you could say, I don't know, like find me 1,000 of these and say accept. The default is 50, assuming that you're just trying to put together maybe like some kind of training data set. If you actually want sort of the bulk of the population, you can pick a larger number. Importantly, the nearest neighbors is not a clustering algorithm used here. What it is ordering all the cells in this population. There we go. There's our 1,000 most similar cells, and you can take a look, and I think we did a pretty darn good job at that, and that most of these have that same, like two linear strands of DNA in the picture. Anyway, so I was in the midst of saying the nearest neighbors will just order the cells from most similar to least similar, and however many you say you want, it takes the top that many. So you could, if you put a very large number, it's possible you will get, some dissimilar cells towards the end of it if you ran out of cells that look the same. But I felt fairly confident within this group that we might find 1,000. It's a 250,000 cell data set. So even if we've gotten down to a smaller portion of M phase, then that's fine. Okay, so there's my similar to anaphase cells that I have collected and put into an image set. And perhaps the next thing I'd like to do is make that a population. So if I hit the make it a population button, we can see we've got a derived parameter similar to anaphase. And then the algorithm is auto gating on that derived parameter to get me the things that are, you know, within my threshold, within my set of cells over here, and the ones that are not. So that is calculating at the moment, we can see. All right. Go calculation. There we go. Similar to anaphase, 0 and 1 are default parameter names, are generally pretty, pretty just generic. You can rename if you want, or rename not anaphase. And maybe anaphase. Okay. And there's 1,000, and there's the 24,000, or sorry, 249,000 that were not in that 1,000. All right. Double click on that, and it works like, any other population in FlowJo. You can go back and put whatever, whatever displays you want on at this point. Well, that makes sense. They're both relatively high for 147 and neon green. Okay. All right. I think, and by the way, I should always mention that you can take these snapshots wherever you want. The snapshot puts that 1,000 image wall into your layout editor. And at any point, let's turn off the image sets. You can take the image wall and add it to your layout editor as well. So if there's some kind of, representative population you want to get into a figure or a poster or whatever, that's a great way to do it. Okay. You know what? I lost track of the chat. I'm going to look at it in a moment just to make sure. All right, cool. We are doing okay with the chat. And let's do the last step that I outlined in that presentation, which was to take that population now, this anaphase population, and turn it into a series of just standard gates so that one might be able to, again, either find more of these cells by applying those gates at a more general level, or probably better, save this workspace and open it up on Chorus to be able to then sort it. I'm going to go to the top level, as HyperFinder always works off the top level. Workspace and HyperFinder. Okay. So I will mention the built-in HyperFinder is better than the plugin HyperFinder. If you still have that one, we built it in and we stopped updating the old one. So this one is significantly better for doing HyperFindery kind of things. You've got a few different kinds of shaped gates that you can make. I think the polytope is generally regarded as the best one. You can set a maximum number of gates if you want. The reason for this was that there were versions of Diva, if people were using HyperFinder with Diva, that had an 8 gate limit. I think that's not true for most modern versions of Diva. You can bump this up if you want, up to you. Um, You've got a downsampling limit here if you want to reduce the total number of events that you're going to work with. By default, it's set to all of them. All right, and then by default, the F measure beta, which is the weight between specificity, oh boy, um, and uh, and whatever, getting them all, getting as many of the, and I just forgot the word, such as life, is balanced there, or one is treat them equally. And then so you'll pick the sensitivity. That was the word I couldn't think of. Geez, definitely getting old. Anyway, so we can pick which parameters are fair game to use. And so right off the bat, we'll probably pick all the fluorescent parameters. We'll pick scatters. Light loss is fine if it wants to use light loss. Um, Generally, you can pick, if you don't want to think about it, just pick a lot of stuff. And, for example, we could just shift and pick all that stuff here. And then go back and maybe pick out a couple individuals. Actually, I could have shifted through most of this too. The only thing that will be a benefit if you pick fewer parameters is it'll go a little faster. So your call. And then last, you will want to tell it which, oh, did I run it in the wrong workspace? Ha, look at me. I'm like, why do I only have two populations? Well, let's close out of this one. We want to find our anaphase population. So we'll do that one more time, where I will pick a very large number of, oh, I still only appear to have two populations. That is weird. It is thinking of the other workspace, I think. Let's make sure I've closed that then. This didn't really need that guy anyway. Okay, I'll make sure that it knows those are there. Yay. Okay, cool. One more time. Pick a whole bunch of parameters. Yeah, I will go to diffusivity and stop with the delta center of mass and then throw in a few different scatters that we could use. Keep it with 8. One's fine. Let us find anaphase. All right, and say OK. So this will pop up this window. It'll tell you in the top right corner it's moving. It will pop this window up if it finds a parameter with no variance. So with the older version of HyperFinder, a no variance plugin would make the algorithm just die. So one of the newer versions, and once you moved it to built-in, just removes any parameter that's got no variance and then proceeds with its building a gate, training a hyperbox, and all their cool sounding stuff, which basically means it's going to divide your target population, my anaphase cells, into two parts, a training data set and a validation set. It's going to draw gates around the green events and see if the red ones fall into it, and then adjust the gates, again, to maximize F measure, precision and sensitivity. And precision and recall, I think, are the proper pairing. Sensitivity and specificity are the other pairing. And so a 1 is a perfect score for an F measure. Right now it's working on improving from a 0.8. So overall, that's pretty good. And we'll see if we can get a little better as it refines the gates and also attempts to reduce the total number of gates to be as simple as possible without impacting the overall F score. So there it goes. And then I always find this pretty cool. It'll pop up a picture of the gates that it used to identify the populations, and it's an algorithm that just finds the best set of gates to get to a population, and it'll frequently pick parameter combinations in orders or combinations that are not something that you or I, as the expert cytometrist, might have done, which is sort of a neat thing to see in the end. All right, we're getting there. And once it's done, picture will pop up. It'll say it's done, and the gates will appear in the workspace all with hi-fi preceding them. And yes, you could have made this go faster by picking less parameters, which takes a little bit of time to do. So, up to you how you want to spend the time. But the dimensionality is down to 9, so there's not too much further. There's not too much lower it can go. All right. Creating prototype projections or polytope prototype projections. You can see which parameters it's using if you have really good eyesight. All right, here we go. So various drawn gates, various sets of parameters. So Alexa floor versus the short axis moment was apparently a good, hadn't even looked at those parameters. A good start. You can see green and red for where it is validated. Our F measure is meh, so maybe I've picked, I don't know, a tough population or lots and lots of parameters, but for the time being, good enough. And we can see that we've got a series of hi-fi gates, and it tells you which parameters it used. So Lex floor, center of mass versus eccentricity of neon green, etc., to get down to our population. 2,464 cells, um, which is nice because I picked out about 1,000 just using the images. So we can actually then, if we want, take a peek at that. Let me turn off regions and edges again and say that's not bad. A couple of iffy ones in there, but almost all of them have that shape that we were looking for. And now we've probably got the right number of anaphase events that were in the total experiment. Okay. This could just be saved. File save. And Chorus can open this file, this FlowJo workspace file, to then be able to use those gates if you'd like, to be able to do sorting on them. Okay. And that is the list of topics I wanted to make sure I brought up today. I'm going to check the chat again. We are empty in the chat. So turn my video back on. So we are at Q&A time. If anybody has questions, I'm watching the chat. And if you have no questions, then I'll give you 10 minutes of your time back. And make sure I look in the actual Q&A. Okay, good. Nothing in the actual Q&A either. There is a chat that just is right there at the top of your screen that's easy to find and see, in a Q&A that you have to go through a more button to get to. So I tend to just like to look at the chat, but making sure everybody has the opportunity to ask questions. And thank you, 30 some folks, for joining me today. It's a nice group. We'll be posting this video to the webpage under previously recorded videos, and I will promote it, I guess, on LinkedIn too, so that people know that there's a spot they can come watch and see most of the tools that are in the CellView Lens pointed out. You're welcome, Melissa. Okay, I guess I will just open this up while we wrap up and just mention there's a bunch of tools under the Tools drop-down menu. I talked about almost all of them either through the menu or through the other places that they're worked on, like the image sets, for example, has a drop-down menu that you can use to get to. It's the same as clicking this button here, but there was a slideshow version of looking at the cells, which can be kind of fun if you want to look at it that way. The various snapshots that I showed to the camera image are there as well. there is a place to export selected cell populations. That's a good one to mention in that I didn't use it in this particular experiment, but this is a place where you can highlight multiple populations and export them, and they'll go somewhere, or populations from different samples, and it'll write the image like a text file that tells you which image goes with which picture. So it allows you to like put together multiple files that would potentially have conflicting image numbers. Like this was image one in multiple files, and put those together for comparison at some later date. There's your extract TIFFs from CW actually as a button, and so on. Generate image file report, get a text file of what images have what cells have what images and what number they are. So a bunch of extra tools in there. I think we hit the most exciting and interesting ones. There is a plate viewer. Didn't have plate data right today, but that's nice to use as well. All right. Well, thank you all for being quite so generous in the comments. That is nice of all of you. Um, I think we're good then. I will give you back a little less than 10 minutes of your time. But thank you for joining me today. Nice to hear it was helpful. Have a great day and a great weekend.